Welcome to all. Here we see about some important common carbohydrates. All we know about that the carbohydrates are biomolecules in organic molecules, and in the sense of that is the macromolecule because the many complex atoms are present in here, so macromolecule. And we consume lot of carbohydrates in available in nature, and we consume through our diet. Some important dietary carbohydrates are disaccharides, polysaccharides, and even monosaccharides. We see about one by one about the some important carbohydrates in this chapter. First we see about the disaccharides. The disaccharides have two monosaccharide unit, and they are held together by special bond that is known as glycosidic linkage. So that disaccharides also known as glycosides. Most common disaccharides of our diet are maltose, lactose, and sucrose. Here first we see about the maltose. The maltose, the structure is based, it contains two glucose units. The anomeric carbon atom of first glucose is connected with the carbon atom of fourth carbon atom of second glucose by the glycosidic linkage. Is The linkage is known as alpha 1,4 linkage. Here the alpha indicate the configuration of anomeric carbon atom of both glucose units, and number indicate a carbon atom involved in the glycosidic linkage. The maltose is one of the reducing sugar because the functional groups are present, and also the anomeric carbon of second glucose is free. Here contains the aldehyde group, and the sources of maltose are maltose mostly present in germinating cereals and in barley. Commercially, we can buy in shops as malt sugar. It is produced from the hydrolysis of starch. Next, we see about isomaltose. The isomaltose also reducing sugar. It contains two glucose units combined in alpha 1,6 linkage. Thus, first carbon atom of one glucose residue is attached to the sixth carbon atom of adjacent glucose unit by glycosidic linkage, and the partial hydrolysis of glycogen and the starch can also produce the isomaltose. The enzyme oligo 1,6 glycosidase present in intestinal juice, that is the gastric juice, can hydrolyze the isomaltose into glucose unit, and in here, the bond is 1,6 linkage. First in maltose have 1,4 linkage. Then we see about the lactose. Lactose is commonly present in milk products. It contains one glucose and one galactose unit. The anomeric carbon atom of galactose and carbon atom 4 of glucose are involved in glycosidic linkage. The linkage is beta 1,4 linkage. So the lactose also a reducing sugar because anomeric carbon of glucose is free in adjacent. That means the second glucose unit. Then sources of lactose includes all the milk products contain the lactose. And next we see about sucrose. Sucrose is commonly called as table sugar. It is the source is sugarcane, and it contains glucose and fructose. The two molecules of monosaccharide, one is glucose and fructose. The anomeric carbon atom of glucose and anomeric carbon atom of fructose are involved in glycosidic linkage. There are no free the reducing end. So the sucrose is considered as non-reducing sugar. Glucose is in the alpha form, whereas fructose is in beta form. In sucrose, hence the glycosidic linkage of sucrose is designed as alpha beta 1 2 linkage. It is a special linkage. And Sucrose is non-reducing sugar because both the functional group of glucose and fructose are involved in glycosidic linkage. There is no free end also, and but one condition. But after the hydrolysis, the sugar are act as reducing agent. Then we see about the sources. Sources include ripe fruit juice like pineapple, sugar cane juice, and honey, carrot roots, sugar beets, sorghum, and etc. Contain the sucrose is the source is sucrose. Next, inward sugar. It is a term used to the inversion of sugar from one monosaccharide unit to other. Sucrose has special optical rotation of plus 66.5 and hydrolysis changes to 19.8 degree. This special change in optical rotation from dextro to levo. Dextro means left to clockwise the rotation. Sorry, levo means left to clockwise rotation, and the right clock rotation considered as dextro sugar. When sucrose is hydrolyzed, is called as inversion. The hydrolysis mixture containing glucose and fructose is called as invert sugar. So one can change to another. Glucose is changed into fructose and the fructose can change into glucose. So that rotation is known as invert sugar. The change in optical rotation on hydrolysis is because of fructose, which is more levorotatory than dextrorotatory glucose. Then we see about the homopolysaccharides. Before we studied about there are three major disaccharides: maltose, sucrose, and lactose. Then we see about polysaccharides. There are two types of polysaccharide in diets. One is homopolysaccharide, next one is heteropolysaccharides. Here we see about some important homopolysaccharides. The homopolysaccharides are normally starch, glycogen, cellulose, dextran, inulin, and chitin. These are all we intake through diet. All these contain glucose as repeating unit, so it is called homopolysaccharide. This homopolysaccharide also known as homoglycans in various areas. Then first we see about starch. It consists of two parts, a minor amylose component and a major amylopectin component. Okay, the minor amylose that is that is treated with the starch is treated with boiling water. 10 to 20 percentage is solubilized. This soluble form is known as amylose. So this is the minor part also. And amylose is a straight chain polymer of glucose unit. The repeating glucose units are linked by alpha 1 4 linkage, and the molecular weight is 4 lakh Dalton or more. And next we see about amylopectin. The insoluble part of that homopolysaccharide, that means the starch, is known as amylopectin, and that absorb water and form paste like gel, but is highly branched molecules. Uh, but the glucose is the repeating unit, and more than 1 million glucose units are present in each amylopectin molecule. The branching points are made by alpha 1 6 linkage. So then here we can stand about first an amylose contain linear chain of glucose, but here the amylopectin is a branched chain of glucose units. Next, we see about the functions of this starch. It is the major polysaccharide present in our food. It is also called as storage polysaccharide because it serves as a reserve food material in plants. It is present in food grains, tubers, and roots like rice, wheat, potato, and vegetables also. Next, we see about the glycogen. The glycogen is called as animal starch because it is only present in animals, that is synthesized inside of animals. It is the reserve carbohydrate in animals. It is stored in liver and muscle only. About 5% of weight of liver is made up by glycogen. Excess carbohydrates are deposited as glycogen. Glycogen is composed of glucose unit joined by alpha 1,4 linkage in straight chain and also alpha 1,6 linkage present in linear chain, branched chain structure. So the glycogen contain linear chain and a branched chain. Then when we remember that alpha 1,4 linkage is present in linear chain, alpha 1,6 linkage is present in branched chain, branching points only. But this repeating unit is glucose. Molecular weight of glycogen is about 5 million. Innermost core of glycogen contain a primer protein called as glycogenide. So it is the act as primer for attaching the glucose units repeatedly. And so glycogen is more branched and more compact than amylopectin. And the function of glycogens are it is the major storage polysaccharide in human body. It is mainly present in liver and muscle. It is also called as animal starch and it is utilized when the fasting time by the process of glycogenolysis. It is the synthesized inside of our body by the pathway of glycogenesis. And this is the structure of glycogen. Next we see about cellulose. Cellulose is the supporting tissue of plants. Cellulose constitute 99% of cotton, 50% of wood, and is the most abundant organic material in nature. We intake through leafy vegetables and the fruits in the form of cellulose. It's made up of glucose units combined with beta 1,4 linkages and it has a straight line structure with no branching points, no branches. Molecular weight in the order of 2 to 5 million. Beta 1,4 bridges are hydrolyzed by the only enzyme cellobiose. This enzyme only in animal present in other animals only and the termite contain this enzyme but that is not present in human digestive system and it is present in so many microbial floras so the that can able to only cleave that 1,4 linkages that means the beta 1,4 linkages. White ant also digests cellulose with the help of intestinal bacteria. So commercially, it is very utilized the cellulose. Next, we see about dextron. They are branched homopolymer of glucose unit with 1,6 bond, 1,4 bond, and 1,3 linkages. They are produced by microorganism. Molecular weight 1 million to 4 million. They not easily go out of vascular compartment. They are used to intravenous infusion as plasma volume expander for the treatment of hypovolumetric shock. It is the polysaccharide present in bacteria. To maintain plasma volume, dextran is used in clinical medicine. Dental plaque is due to dextran synthesized from sucrose by oral bacteria. Then we see about inulin. Inulin, it is a long chain homoglycan composed of D-fructose unit with repeating beta 1,4 linkages. It is the reserve carbohydrate present in various bulbs and tubers such as chicory, dahlia, dandelion, onion, garlic, etc. Clinically, it is used to renal clearance. It used to find the renal clearance value and the glomerular filtration rate. And the inulin and insulin are different structure. Insulin is an hormone. It is involved in the glucose metabolism and it regulates the glucose in our body. The inulin is a polysaccharide. It is used to determine the glomerular filtration rate in kidney. Then we say about chitin. It is a polysaccharide composed of N-acetyl glucosamine. That means the glycoprotein also glycosidic linkage of beta 1,4. Important structural polysaccharide of invertebrates. crabs, lobster, and insects. So we intake through the seafood. Next, we intake some of the heteropolysaccharides. First in this list, agar. Agar it is prepared from seaweeds, so it contains galactose, glucose, and other sugars. It is dissolved in water at 100 degrees centigrade. It is cooling set into gel. Agar cannot be digested by bacteria and hence used widely as a supporting agent to culture bacterial colonies. Agar is used as a supporting medium for immunodiffusion and immunoelectrophoresis. Agarose is made up of galactose. It's a fine mass of agar combined with 3,6 anhydrogalactose unit. It is used as matrix for electrophoresis. Electrophoresis is an instrumental technique. It is used for the separation of DNA, RNA, and some of the protein. It is used in biochemical laboratories. Next, another one of the heteropolysaccharide is mucopolysaccharides. Mucopolysaccharides are glycosaminoglycans, shortly GAG, are containing uronic acid and amino sugars, and the acetylated amino groups, sulfate, and the carboxyl groups are also generally present. Because of the presence of these charged groups, they attract water molecules and acetylated amino groups, sulfate and carboxyl group also generally present in mucopolysaccharide. And presence of these charged groups, they attract water molecules and so they produce viscous solutions. So that is hemi-solid in structure. Mucopolysaccharides combination with the protein form mucoproteins. Example hyaluronic acid, heparin, chondroitin sulfate, dermatan sulfate, and keratan sulfate. These are present in our body. Then they excreted in urine abnormal amount in group of lysosomal storage disorder known as mucopolysaccharidosis. Next we see about heparin. It is an anticoagulant widely used in laboratories and used in clinically used when taking blood in vitro for clinical studies. It is also used in vivo in suspected thromboembolic condition to prevent intravascular coagulation. Heparin mostly present in liver, lungs, spleen, and monocytes of our body. Commercially, it is prepared by animal lung tissues. It contains repeating unit of sulfated glucosamine, alpha 1,4-L iduronic acid, and so on. Next, we see about chondroitin sulfate. It is present in ground substance of connective tissue, widely distributed in cartilage, bone, tendons, cornea, and skin. Composed of a repeating unit of glucuronic acid, beta 1,3 N-acetyl galactosamine sulfate, beta 1,4 and linkage, and so on. Next, keratin sulfate, it is the only GAG which does not contain any uronic acid. The repeating unit are galactose and N-acetyl glucosamine in beta linkage. It is found in cornea and tendons. In this chapter, next we see about dietary fiber related to the common carbohydrates. Dietary fiber is the name given collectively known as indigestible carbohydrates. That means we consume so many carbohydrates, but we can't digest it through our gut. So some of these are cellulose, pectin, gums, mucilages, hemicellulose, lignin, etc. The dietary fiber is not digested by the enzyme of the human gastrointestinal tract, where most of the other carbohydrates like starch, sugars are digested and absorbed. Plant food are the only sources of dietary fiber. It is found in vegetables, fruits, and grains. Importance of fiber. First, we'll see about water holding capacity. The dietary fibers have the property of holding water and swell like a sponge with a concomitant increase in viscosity. So it hold water in our body, and absorption of organic molecules. In another way, organic molecules like bile acid, neutral carcinogens, and toxic compounds can be absorbed on dietary fiber and facilitates its excretion. And next we see about it increases stool bulk. The fiber absorb water and help to reduce the tendency towards constipation by increasing bowel movements. Next, hypoglycemic effect of fiber. Gum present in fenugreek seeds most effective in reducing blood sugar and cholesterol levels. Uh, Almost 40 percentage is occupied with gum. Hypocholesterolemic effect of fiber. Fiber has cholesterol lowering effect, so that is known as hypercholesterolemic effect, and significant in medicine. It associated with reduced incident of number of diseases like coronary heart diseases, colon cancer, diverticulosis, hemorrhoids, etc. And adverse effect, the nutrition in binding some mineral elements and preventing their proper absorption. So it prevent the proper absorption and lead to deficiency of mineral elements. Carbohydrates are the major source of energy for man. For example, glucose is used in the human body for energy production. It give instant energy for us. It give ATP for its energy currency. Also, it is known as fuel of our body. Some carbohydrates serve as reserve food material. So some of the, for example, the glycogen in animal tissue, starch in plants serve as storage form of food in the form of starch stored in plants and animals in like granules. And the carbohydrates are components of several animal structure and plant structures, and are components of skin, connective tissue, tendons, cartilage, and the bones. And in plants, the cellulose is a component of wood and fiber. Some carbohydrates are component cell membrane and the nervous tissue. Carbohydrates are the components of nucleic acid and blood groups, substances involved in cell to cell interaction, and it is like some of the derivative of carbohydrates are used in drugs. Glycosid or being is used in clinical medicine, and streptomycin, an antibiotic, is a glycosid. Amino sugar derivatives of carbohydrates are components of antibiotic like erythromycin and carbamazepine. Ascorbic acid, a derivative of carbohydrate, is water-soluble vitamin. Bacterial invasion involves hydrolysis of mucopolysaccharide.